students, welcome back to my channel Learn Microbiology. We are doing Unit 1 revision, Lipid Metabolism and Catabolism of Hydrocarbons. So far, we have finished 3 topics in this particular unit. Today, we are going to do the last topic, that is action of lipases on triglycerides tripalmitate. Let's get started. What are triglycerides? In the previous videos, we understood that triglycerides or triacylglycerols are glycerol molecules esterified to 3 fatty acid groups. And one more important thing that we learned was triglycerides are absent in prokaryotes. That is, they are absent in bacteria, but you can find triglycerides in human beings, in animals, in plants, as well as in every eukaryotic cell as a reserve material. Let's today discuss about these triglycerides as well as the action of lipases in this video. Starting on to eukaryotic cells. Eukaryotic cells contain this particular triglycerides as oily droplets in the aqueous cytoplasm, and these oily droplets containing triglycerides will serve as a depot. Depot means it's like a dam or a reservoir of metabolic fuel. It means this particular eukaryotic cell, as we can see here, these particular molecules here are nothing but oily droplets containing triglycerides. So whenever the cell requires energy, it can use this triglycerides for it. Secondly, if you see in vertebrates like human beings, we have a specialized cells called as adipocytes. Now these adipocyte cells, as you can see in this image, will be storing the triglycerides. Thirdly, in plants, triglycerides are stored as oils in seeds, and this will become a precursor for seed generation, seed germination when it needs energy. As we can see in this diagram, you can see over here, this is castor oil seeds, and these castor seeds are containing the triglycerides in the form of oil. Similar we can find in case of sunflower oil, groundnut oil, etc. Let's talk about more about this triglycerides in human beings. Humans have these fat tissues which are called as adipocytes for storing triglycerides under the skin, in the abdominal cavity, as well as in the mammary glands. As we can see in this image over here, I will zoom it for you. We can see this is the outer epidermis, then we have an inner dermis layer, and then you can see over here, these are the fatty tissues under the skin, and these are called as adipocytes tissue. Human beings can store at more than 20 kgs of triacylglycerols. If you take a moderately obese person who is having around 15 to 20 kilograms of triacylglycerols, these people can meet their energy needs for months. It means that this person, if they go without eating for a month, they can easily survive. Moving ahead, let's learn why triacylglycerols or these triglycerides are used as stored fuels, or what are the advantages of using this triacylglycerol as stored foods. The first advantage is the carbon atoms of fatty acids are more reduced. We have seen that all the carbon in this particular fatty acids contains hydrogen. So whenever there is lot of hydrogens in the molecule, that molecule is highly reduced. Because of that, when you undergo oxidation of this triacylglycerol, that is breakdown of this triacylglycerol, this will give you twice amount of energy as compared to the oxidation of carbohydrates. Let's see this example. If you take 1 gram of carbohydrate and you get 4 calories of energy on the oxidation, then for 1 gram of fats or 1 gram of triacylglycerols, you can get around 8 to 9 calories of energy. Isn't it very interesting? Let's move to the second advantage. Triacylglycerols are hydrophobic. Hydrophobic means they are not water loving. Secondly, they are unhydrated. Unhydrated, again, they do not require water around them. Hence, these organisms who are storing these triacylglycerols as their stored food do not need to carry the extra weight of this water of hydration. Now, this water of hydration plays a very important role when we consider another molecules like polysaccharides, because for 2 gram of you require 2 gram of water of this hydration for 1 gram of polysaccharides. So, if you want to store polysaccharides, then for 1 gram polysaccharide, you require 2 gram water. So, see how much extra weight that particular organism has to carry. But if that organism is storing triacylglycerol, it does not require to carry this water of hydration. So, these are the two important advantages of using triacylglycerols as stored fuels. Moving ahead, now we have seen that one of the main important function of triacylglycerol was as stored food. Now, this triacylglycerol also plays some other important roles in some animals. Let's take the examples of polar animals like seals, walruses, and these penguins. You can see them, they are so over here. Those cute penguins and seals you can see over here. Now, these organisms have this particular triacylglycerol under their skin. And what is the function of this triacylglycerol? This is going to give them insulation against low temperature. As we know, these organisms are living under polar conditions. Hence, in that low temperatures, this particular triglycerides can give them insulation. Another important factor or important advantage is, have you heard about hibernating animals like those bears? Now, this bear, it undergo a particular period of time where it hibernates itself. That is, it is not going to eat anything, it is not going to drink any water, but it is going to be alive, it is going to metabolize everything. So, how this animal can do this? Because this animal has a huge fat reserve this animal has a huge fat reserve on its body, and this particular fat consisting of triglycerides, it provides insulation as well as it is important for its energy. So, what all months this particular bear is going to go for this hibernation, the entire energy comes from this stored triglycerides. The third important advantage here is, as we know, triacylglycerols are usually oils, they have a very low density. See, I will take an example. If you take your water and if you add few drops of oil, we know that oil will float on the water. The reason is it has a very low density. Now, this particular property of triacylglycerol is serving the important function of these sperm whales. Now, the sperm whales, when they go deep into the sea, where there is very low temperature, and over there it just going to float, or not. Float is not a correct word, but it is not going to swim. So that is its feature. So how this organism can just stay over there? It can only do so when it will match the buoyancy of the water as well as its own body. When the both buoyancy, or in simple terms density, matches, only then it will be able to do so. So these sperm whales, on their head there is a particular organ to store this triglyceride in the form of spermaceti oils. Now, this spermaceti oil, when this particular whale go into the deep ocean where there is a low temperature, it will get solidified, and that's how this particular whale can match the buoyancy of the water at the underwater level. But once the whale, when it comes back on the surface, because of the increase in temperature, this spermaceti oil will go and melt, its density will change, and now it will match that of the water on the surface. Isn't this interesting facts about triglycerides? If you want to read more on this hibernating birds as well as on this sperm whale, I have given some particular here. There is some article which is taken from Leninger book itself. You can just pause the video and read this for your extra information, and even I have it for your wear as well. Let's move ahead. So far we understood what are lipids, what are its classification, what are its types, and also its main functions, which type of lipids are present in prokaryotes, which type of lipid is present in eukaryotes, so on. Now, as we studied right now, that this triglyceride is going to be stored in the particular body for energy purpose in the future. Now, how this particular triglyceride will give energy in the future? That is a question. As we can see, this triglyceride. So, what happens is, this adipocyte tissues and this germinating seeds which contain the triglyceride, they also contain one enzyme which is very important called as lipases. Now, what the lipase enzyme does? Lipase enzyme is going to break the ester bond. I hope you remember that there was an ester bond formed between our glycerol and the fatty acid. Yes or no? So that particular you can see such type of bond here, and that bond will be broken down by this action of this lipase enzyme, wherein it will release your fatty acids free. Now, when these fatty acids become free, then they can be exported to where all said it is required, and then it can be further metabolized to give energy. So, I hope this whatever I just said right now, it is not in such a manner that you will understand. So, I have created a short animation for you so that you can understand this better. Can you all see this lipase enzyme over here? Okay. Now this lipase enzyme is going to go and break the ester bond. As we can see, it went there and it's broken the ester bond, and that is the reason now your glycerol molecule and your fatty acid molecules have been separated. Secondly, it is going to add water over those bonds. If you remember, students, that there used to be removal of water when this bond was forming. So when now this bond was getting broken down, that water comes back, and now that water is getting added. Once water is added, it will split to form its HOH, that is hydrogen and hydroxyl, and we can see we have separated our glycerol with these three fatty acids. So that is the action of lipase enzyme. It will break the ester bond and it will release free fatty acids and your glycerol molecule. Now this glycerol molecule is ready to load new fatty acids, correct, to form lipids, while this particular fatty acid will leave the cell here and will go to where all it is required. For example, if you see human beings, we studied that in abdomen as well as in below the skin, this particular triglycerides are stored in adipocytes, correct? But now if you're going, imagine you're going into let's say fasting for a few months, or let's say let's not go for months, let's take 1 week. Okay, so now you have to completely rely on the stored food. So now when this has to happen, the lipase enzyme in this adipose tissue will get activated. It will break the ester bond and it will release this free fatty acids. Now these free fatty acids will come out of this adipocytes. It will be transported to all the cells of your body, and once it will enter the body, it will undergo further catabolic reactions, break down to give you energy. In the coming units, we are going to study how this fatty acids is broken down further to release energy. So we can see the entire hero of our reaction is our lipase enzyme. So don't forget. So far we have finished our 1.1 introduction to lipids. All the 4 topics have been covered. If you have missed any video, you will find its link in the description box. Go ahead and find out. The entire topic which we covered today is from your Leninger Principle of Biochemistry book. Thanks for watching. Subscribe to my channel, as also click this bell icon so that you will be notified whenever I post a new video. See you all in the next video.